Welcome back to Biochemistry Scientists. Today we're going to be covering the first half of Chapter 17 in your book, which is glycolysis. The two objectives for this portion of the chapter are one, to describe the overall purpose of glycolysis, including its reactants and products, where it's localized in the cell, and its tissue distribution. And then the second concept I want you to be able to explain is substrate level phosphorylation and why it's important. Glycolysis is the first stage of glucose metabolism in the cell, and it's actually divided into two phases. In the first phase, one glucose molecule is converted to fructose 1,6-bisphosphate, and then this will give rise to two molecules of pyruvate. And once pyruvate is formed, it has one of several fates that we'll be talking about in the next video. But this reaction plays key roles in the way organisms extract energy from nutrients, so this is why we focus so much on glycolysis. In the first phase of glycolysis, there is an energy input, and as you can see here, ATP is broken down into ADP, and this energy input is required to drive the first few stages of glycolysis. And then in the second phase, you have ultimately an energy output, where 4 ATP molecules are produced and NAD is reduced to NADH. Ultimately, the final product of glycolysis is to make pyruvate. And then pyruvate has 3 different fates. In contracting skeletal muscle or other types of muscle where oxygen supply is low, the cell goes through anaerobic metabolism and they end up producing lactate. However, in other tissues where there's plenty of supply of oxygen, the cell will go through oxidative phosphorylation, or actually they'll go through a citric acid cycle followed by oxidative phosphorylation. But you need oxygen for this to occur. And finally, in fermenting yeast, this doesn't actually occur in humans, they can go through anaerobic alcoholic fermentation. So instead of taking pyruvate and turning it into lactic acid, like contracting muscle, they turn it into ethanol. And I talked about these two phases of glycolysis. The first phase is a preparation phase, in which a 6-carbon glucose molecule is broken down into 2 3-carbon glyceraldehyde 3-phosphate molecules. And this actually requires an input of energy in the form of ATP. Let's talk about each of the reactions in phase 1. In step 1 of phase 1, glucose is phosphorylated to give glucose 6-phosphate. This reaction is an endergonic reaction, meaning it requires an input of energy, and it's actually driven by the free energy of hydrolysis of ATP. So when ATP is hydrolyzed, energy is released, and that's harnessed in order to drive the endergonic reaction. I want you to see here that I've numbered the carbons for you. So that's the number 1 carbon on glucose and glucose 6-phosphate, and the number 6 carbon on each. So this is why we call it glucose 6-phosphate, because it's still a glucose molecule with a phosphate group added to the number 6 position. The enzyme that catalyzes this reaction is a hexokinase. So a hexokinase, from the name, you can tell that first, it adds a phosphate group. That's what a kinase enzyme does. And second, it works with any hexose. So it doesn't have to just be glucose. It's any 6-carbon sugar molecule. In the liver, there is an enzyme called glucokinase. It's a form of hexokinase that's specific to glucose. This actually follows the induced fit model that we learned about earlier for enzyme reactions, which is pretty cool because there's a X-ray crystallography research to show that when glucose and ATP bind to this enzyme, there's a conformational change in the enzyme that seems to increase its enzymatic activity. The second step in this phase is isomerization of glucose 6-phosphate to produce fructose 6-phosphate. And the enzyme that catalyzes this is glucose phosphate isomerase. Makes sense, right? And so basically what you see is that the aldehyde on the C1 of glucose 6-phosphate is reduced to a hydroxyl group, and the C2 hydroxyl group is oxidized to give the ketone group on the fructose 6-phosphate. In step 3, fructose 6-phosphate is phosphorylated again to generate fructose 1,6-bisphosphate. So this is the second reaction that is coupled to ATP hydrolysis, and the enzyme that catalyzes this is phosphofructokinase. So what you see here is that a second phosphate group is added to the carbon at the number 1 position. So now we have 2 phosphates. So we name this second molecule for where the phosphates are located, right? So you can see fructose 1,6-bisphosphate. Phosphofructokinase exists as a tetramer and it's subject to allosteric feedback. The tetramer is composed of L and M subunits, and there's all different versions that exist. Combinations of these subunits are called isozymes. Muscles are rich in the M subunit, and the liver one is rich in the L subunit. So ATP is an allosteric effector of this enzyme, which means that high levels of ATP inhibit the enzyme while low levels of ATP activate it. The other thing you should know is that fructose 1,6-bisphosphate is also a key allosteric effector of this enzyme. So what that means is production of the F1BP is going to inhibit the effect of that enzyme. Moving on to step 4, what you see is that fructose 1,6-bisphosphate is split into two 3-carbon fragments. One is called dihydroxyacetone phosphate, and the other is glyceraldehyde 3-phosphate. And the enzyme that catalyzes that reaction is called aldolase. And so I'm labeling the carbons for you to see here so you see how that 6-carbon molecule is split into two 3-carbon fragments. The final step in that first phase of glycolysis causes the conversion of dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So now we actually have two glyceraldehyde 3-phosphate molecules, one produced in step 4 and then the other produced in step 5. The enzyme that catalyzes this reaction is called triose phosphate isomerases. And both of these compounds, you see they have 3 carbons, therefore they are trioses. This reaction actually has a small positive delta G, but what you're going to need to learn is that glycolysis has several reactions that have very negative delta Gs. So what they do, those negative delta G reactions tend to drive the reactions that have a positive delta G so that the overall process is going to be negative. And we're going to look more at the delta Gs when we do the second half of this chapter. In summary, the first phase of glycolysis, glucose, a 6-carbon molecule, is converted into two 3-carbon glyceraldehyde 3-phosphate molecules. And what's important to note is that the key intermediate in this series of reactions is fructose 1,6-bisphosphate. And the enzyme that catalyzes this reaction, phosphofructokinase, is subject to allosteric control. Why is that important? Basically, it's the cell's way of detecting whether or not you're going to need more glucose broken down or not. So when ATP levels are high, it's going to inhibit phosphofructokinase activity, and when ATP levels are low, it's going to stimulate phosphofructokinase activity, basically upregulating this phase of the reactions. So for phase 2, you have another 5 sets of reactions, and what you see is that glyceraldehyde 3-phosphate is converted into pyruvate. The thing that happens here is that it occurs in duplicate because recall we made 2 glyceraldehyde 3-phosphate molecules for every 1 glucose molecule. And this phase of the reaction is going to have energy output in the form of ATP and NADH. In the step 6, you see that glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. So what's happening here is that a phosphate group is added to the first carbon in this molecule. And the enzyme that catalyzes this reaction is called glyceraldehyde 3-phosphate dehydrogenase. What I want to point out here are a couple things. First, the phosphate that's added does not come from ATP, and this is different than all the other reactions we've seen so far. It comes from an inorganic phosphate. And then the second thing I want you to point out here is that NAD is reduced to NADH. And so this is why it's called a dehydrogenase molecule, because we're removing that hydrogen in the process of reducing NAD. The conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate can be subdivided into 2 half reactions. First, you have the oxidation of glyceraldehyde 3-phosphate, and this reaction is exergonic, meaning it has a negative delta G. And then the second reaction is actually endergonic. It has a positive delta G, so that overall the reaction has a small positive delta G. And the second reaction is the phosphorylation of 3-phosphoglycerate to make 1,3-bisphosphoglycerate. The enzyme that catalyzes this reaction is glyceraldehyde 3,5-phosphate dehydrogenase, which is actually a class of NADH-linked dehydrogenases. At the active site of this enzyme, it binds an NAD+ molecule and contains a critical cysteine residue. So reduction of the NAD to NADH causes it to form an enzyme thioester intermediate, so that in the second step, the phosphate ion attacks the thioester derivative of glyceraldehyde 3-phosphate dehydrogenase to produce the final product, 1,3-bisphosphoglycerate, and that's going to then regenerate the thiol group of that very important cysteine at the active site of the enzyme. So why do we care? When that phosphate group is added to make 1,3-bisphosphoglycerate, there's a high energy bond there that is going to be able to drive substrate level phosphorylation that's catalyzed by phosphoglycerate kinase in the next reaction. So what do I mean by substrate level phosphorylation? So what that is it takes the phosphate from one molecule and adds it to an ADP to make ATP. And in the future, we're going to learn about oxidative phosphorylation that occurs in the electron transport chain. So this is very different. This is going to be taking that phosphate from a molecule and adding it to ADP. Ultimately, the sum of the endergonic phosphorylation of ADP and the exergonic hydrolysis of the mixed phosphate anhydride are what's going to give you the delta G for this reaction. Step 8 is an isomerization reaction. So you see this enzyme phosphoglycerol mutase that basically moves the phosphate from carbon number 3 to carbon number 2. Step 9 is catalyzed by enolase. So basically 2-phosphoglycerate loses a water molecule to produce phosphoenolpyruvate. And what I want to point out here is that phosphoenolpyruvate contains a high energy bond to that phosphate group, which is going to be important in the final reaction. And why is that important? Because phosphoenolpyruvate transfers its phosphate group to ADP, finally producing ATP and pyruvate. The enzyme that catalyzes this is pyruvate kinase, and it has a delta G of hydrolysis of phosphoenolpyruvate that's more than the hydrolysis would be for ATP. So you can see it has a very high energy bond. The other thing I want to point out with this reaction and quite a few other reactions is that you're going to see magnesium ion showing up as a cofactor. So this is a factor that is not modified by the reaction, but it's important in catalyzing the reaction. In the final phase of glycolysis, you're going to see that 2 molecules of pyruvate are produced for every molecule of glucose that enters the pathway. And these are going to be able to provide energy for other reactions in the cell because they involve electron transfer. So you see that NAD is reduced to NADH, and it's going to involve the production of 2 ATP molecules. So in the first stage, we have an input of energy in the form of ATP, and in the second phase, we have an output of energy, so that the net final production of ATP is actually 2 ATP molecules for each glucose molecule. Let's quickly review the learning objectives for this section and then go on to talk about homework. First, I want you to be able to describe the overall purpose of glycolysis, reactants, products, cellular localization, and tissue distribution. So what I want to know for reactants and products are the names of the reactants and products, the enzyme that catalyzes the reaction, and you need to start making note cards so you can identify the products. Obviously, I'm not going to have you draw them out because I can't online, but you are going to need to be able to identify the products and give me the name if you see a figure. Second, explain the concept of substrate level phosphorylation. You should go back and review those slides. I've explained it in pretty big detail, but I need you to understand how it works and why it's important. That's it for this part of Chapter 17. You have some homework assigned. An answer key will be posted for that, but you should try and do it on your own, and it will be due after Exam 3. However, in order to review for Exam 3, it would be a good idea to do the homework first. And speaking of Exam 3, it will be on Chapters 12, 15, 16, and 17. Finally, I'm going to scan the page that has the homework so that you can see if your book has the same question numbers as mine. All right, happy studying. Don't forget to make those note cards on glycolysis.